Hello everyone, welcome back to the session in metabolism. Here we will be dealing with the beta oxidation fatty acids. We'll be continuing with the second step. We already saw that the oxidation, it takes place in three stages. The first stage where the fatty acid is getting activated to fatty acyl coenzyme A, and then this is happening in cytoplasm, and then this fatty acyl coenzyme is getting transported through the mitochondrial membrane to reach the mitochondrial matrix, where the beta oxidation proper, the reactions are going on, because the enzymes which are required for the beta oxidation process, it is present in the mitochondrial matrix. Okay, so here you can see the activation of fatty acid. It has already been dealt in the previous presentation. We will here look into the second and the third step. Okay, the second step is Carnitine shuttle. Okay, so fatty acyl coenzyme A, which is being produced in the cytoplasm, has to be transported to the mitochondrial matrix. Now, what happens is the outer mitochondrial membrane is not a hurdle for the transportation, but inner mitochondrial membrane is obviously it needs like the fatty acyl coenzyme A, it needs a carrier protein or a transportation system for it to cross the inner mitochondrial membrane, and that is where the carnitine molecule comes up. Okay, carnitine mediated shuttling, it takes place that so that the fatty acyl coenzyme A, it can get transported through the inner mitochondrial membrane and reach the mitochondrial matrix. So how it happens now? Before that, what is actually carnitine? Carnitine, it is a zwitter ionic alcohol. Chemically, if we see, it is beta hydroxy gamma trimethyl ammonium butyrate. Okay, leave those parts. It is an alcoholic molecule, a zwitter ionic alcohol, and you can see here the fatty acyl coenzyme A, during the carnitine shuttle, it reaches the outer surface. So this is the inner mitochondrial membrane, isn't it? The carnitine, it is found on the outer surface of the inner mitochondrial membrane. Okay, now the fatty acyl coenzyme A, it binds with the carnitine. Okay, and you can see this reaction is a reversible reaction. So the fatty acyl coenzyme A, it binds with carnitine on the outer side of the inner mitochondrial membrane. Okay, and this reaction, it results in the formation of fatty acyl carnitine, and during this process coenzyme A is released. Now, this fatty acyl carnitine is the one which is going to get transported through the inner mitochondrial membrane. The whole reaction, it is catalyzed by the carnitine acyl transferase 1, or we can say CAT1, CAT1. Okay, now the fatty acyl carnitine 1, it can cross the inner mitochondrial membrane to reach the mitochondrial matrix with the help of a carrier protein. And this carrier protein, it is nothing but the carnitine translocase enzyme. Here you can see the membrane, isn't it? It is a carnitine translocase. Translocase in the sense like changing the location, isn't it? So here from cytosol to the mitochondrial matrix, that is what the carrier protein is doing, isn't it? So here it is carnitine translocase, that is the carrier protein. So it helps in the facilitated diffusion of the acyl carnitine from the outer surface of the inner mitochondrial membrane to the mitochondrial matrix. Okay, so now acyl carnitine, it moves through the inner mitochondrial membrane, and it reaches the inner side of the inner mitochondrial membrane. But reaching over here, what happens is the carnitine has to be released, and fatty acyl compound has to be like changed back into acyl coenzyme so that it can enter the beta oxidation proper. And this is taking place with the help of the next enzyme. It is CAT2, carnitine acyl transferase 2. So what happens is the acyl carnitine, it combines with the coenzyme A, and it results in the formation of fatty acyl coenzyme A. So the fatty acyl coenzyme A is regenerated here, and carnitine is regenerated again. Okay, it releases carnitine, and this carnitine, it will get transported back to the outer surface of the inner mitochondrial membrane so as to be used for the transportation or translocation of a new fatty acyl coenzyme. Okay, now this transportation of carnitine back to the outer surface is also done with the help of the carrier protein carnitine translocase. Okay, now what happens here? Okay, this fatty acyl carnitine, it combines with coenzyme A and produces fatty acyl coenzyme A and carnitine. And this is also done by another carnitine acyl transferase, specifically the CAT2. Okay, so this is about the carnitine shuttle, where the fatty acyl coenzyme A, it gets transported across the inner mitochondrial membrane to the matrix with the help of carnitine, carnitine translocase carrier protein, and carnitine acyl transferase 1 and 2 enzymes. Okay, I hope it is clear. Now, the fatty acyl coenzyme A is present in the mitochondrial matrix. Now, it can enter the beta oxidation proper. So, this is the beta oxidation proper, which is taking place in the mitochondrial matrix. Now, if you look into the beta oxidation proper, it takes place in 4 steps. Okay, the first is oxidation, dehydrogenation or oxidation. Second step, it is hydration. Third, it is dehydrogenation or oxidation again. Fourth is thiolysis or thiolytic cleavage. Okay, we'll see step by step. The first step is oxidation or dehydrogenation, right? So this is a fatty acyl coenzyme which has reached the mitochondrial matrix. It undergoes dehydrogenation, that is, it is oxidation itself, right? So H2, it is being released from fatty acyl coenzyme A, and during this process, double bond is introduced. Now, so double bond is introduced between these two. So this is the first carbon. You have to number it from this side, isn't it? This is a functional group. So first carbon, second carbon, third carbon. So it is between the second and the third carbon. Okay. Since this is a functional group, we call this as alpha carbon and beta carbon, and continuing gamma and likewise. Okay. So double bond is produced as a result of dehydrogenation, and this double bond is created between the alpha and the beta carbon, or the second and the third carbon. Okay, and this undergoes dehydrogenation with the help of the oxidizing agent FAD, the coenzyme FAD, which gets reduced to FADH2. Okay, so FAD is the electron acceptor, or we can say the hydrogen acceptor. Okay, and what is produced? It is a enoyl compound, because it is a double bond over here, so it is enoyl coenzyme, trans-enoyl coenzyme. Okay, a double bond is produced. Now this trans-enoyl coenzyme, you can see here that is a first reaction, fatty acyl coenzyme getting dehydrogenated to produce trans-enoyl coenzyme. Now what does this indicate? This indicates the double bond where it is present. It is on the second carbon. Okay, so it is on the second carbon between the second and third. Okay, there is only one double bond. That is why there is only one number. If there were more, you could have found 2 comma the next number, etc. Okay, coming back, it is trans-enoyl coenzyme. Now the trans-enoyl coenzyme, it undergoes hydration. That is, a water molecule is added to the double bond over here. Okay, so as a result, you can see a hydroxyl compound is produced. So hydroxyl group is added onto the third carbon here. Okay, so what is the product produced? It is 3-hydroxy acyl coenzyme. Okay, it is just like this one, but only one thing is, instead of hydrogen over here on the third carbon, it is hydroxyl group. So the name changes to 3-hydroxy acyl coenzyme. And this reaction is catalyzed by enoyl coenzyme hydratase. Okay, now the 3-hydroxyl coenzyme A, produced as a result of hydration, it undergoes dehydrogenation, or it is the oxidation process. So this is the second oxidation step, or second dehydrogenation step. Okay, now here it is the NAD plus is the hydrogen acceptor, or it is the oxidizing agent, as a result of which the hydrogen atoms, 2 hydrogen atoms are being released, and NAD plus, it gets converted to NADH, and a free hydrogen ion is being produced. It is released. Okay, now what happens to 3-hydroxy acyl coenzyme A? The 3-hydroxy acyl coenzyme A, it gets converted into beta keto acyl coenzyme A. Why it is beta keto? Because it is beta carbon is in the keto form. Okay, it is fatty acyl coenzyme A itself. Only the difference is beta carbon is in the keto form. So we have beta keto acyl coenzyme A. Okay, how is beta ketoacyl coenzyme produced? It is by the dehydrogenation of 3-hydroxyacyl coenzyme. The reaction is catalyzed by 3-hydroxyacyl coenzyme dehydrogenase. It is very easy to learn the names, isn't it? Because 3-hydroxyacyl coenzyme is the substrate. It undergoes dehydrogenation, and hence the enzyme 3-hydroxyacyl coenzyme dehydrogenase. Okay. Now, the last on the fourth reaction or the fourth step, it is the thiolysis. Okay. Here what happens is a coenzyme is being used here. Okay, so beta ketoacyl coenzyme, it combines with or reacts with a molecule of coenzyme and split off the and split off to produce or to release acetyl coenzyme. So how it is being produced? The added coenzyme A, from there the hydrogen over here, it is getting added to here. Okay, so so a bond is broken over here, and the whole coenzyme A, we can see the hydrogen is added here, so it has become CH3COSCOA, which is acetyl coenzyme. Okay, and it becomes C double bond O SCOA. Okay, so it is another acetyl fatty acyl coenzyme. Okay, now what is the difference of this fatty acyl coenzyme and this one, the initial one? The initial one had, you can see here, what you call CH3, CH2N, then again 3 carbon. Okay, 2 carbon and what you call the C double bond or SCOA, right? But over here, what has happened? Those 2 carbons have been lost or have been released as acetyl coenzyme. Okay, so we can see that the fatty acyl coenzyme, which is produced from this particular substrate, is actually 2 carbon less in length. Okay, so the length of the fatty acyl coenzyme produced after beta oxidation is 2 carbon less in length. Okay, now this fatty acyl coenzyme, it can again enter the beta oxidation, and it can take undergo oxidation, hydration, second oxidation, thiolysis, and produce a molecule of acetyl coenzyme and another fatty acyl coenzyme, which is having length 2 carbon less than this one. Okay, so this can continue till we have the complete oxidation is over. That is, finally what we will have is a fatty acyl coenzyme will split into 2 acetyl coenzyme. So till then this reaction can continue. Okay, so that is about the beta oxidation proper. I hope this is clear. Now we'll see the with an example, right? So this is the summary. A fatty acid, it has undergone activation to produce acyl coenzyme, and with the help of translocase enzyme and the carnitine shuttle, it enters the mitochondrial matrix, where the fatty acyl coenzyme, it undergoes oxidation, it undergoes hydration, right? Enoyl compound, right? And then hydroxy compound is produced. It undergoes oxidation, the second oxidation, and then they have the cleavage, thiolytic cleavage. Okay, acetyl coenzyme is produced. This acetyl coenzyme can enter the Krebs cycle, isn't it? And it can contribute to the ATPs being produced. Okay, now this is one example. It is C17 molecule. You can see here it is 1 carbon over here, 14 carbon. Then you have one more carbon, isn't it? So 14, 15, 16. Okay, so here we have this. You can see how many. Yeah, so it is C16 compound, the fatty acid. How many acetyl coenzyme are produced from C16 fatty acid? C16 fatty acid will give rise to 8 acetyl coenzyme, right? Along with this, 7 FADH2, 7 NADH are being produced, right? And I hope you remember during the acetylation or activation, one ATP molecule is being used up, right? So it is minus 2. So altogether what happens is 7 NADH is produced, 7 FADH2 it is being produced. At the same time, ATP molecule is being used up. Okay, for a molecule of fatty acid with what we call 16 carbon. Now here you can see the